In the previous video, we talked about stromal cells and the extracellular matrix in the bone marrow microenvironment. And I'll put a link to that video in the description of this one just in case you missed it. I briefly touched on what hematopoiesis is, and I'm going to talk about the different characteristics of granulopoiesis, which is the hematopoiesis of eosinophils, basophils, and neutrophils. I'm also going to talk about monocytopoiesis, which is the hematopoiesis of monocytes, macrophages, dendritic cells, and then lymphopoiesis, which is natural killer cells, natural killer T cells, and B cells. So hematopoiesis is just the creation, creation or the differentiation of a hematopoietic stem cell, which is what I discussed in the previous video, that differentiates into one of these other types of white blood cells for an immune response. So granulopoiesis, some of the regulatory factors are interleukin-3. So and then you have GM-CSF and G-CSF, sorry. And a little bit of the characteristics of its maturation would be nuclear segmentation and then acquisition of primary then secondary granules. So if you look at something like this massive stem cell here with its nucleus, it might go down here and shrink a little bit, and you're going to have a kind of weirder looking nucleus here. And then here, you got an even smaller one with a smaller nucleus like this. And then finally here, you have the smallest one with a nucleus that is very segmented like this. And the production time of a granulocyte is anywhere from 5 to 7 days. So I'll write that right here. So 5 to 7 days for that. The rate of formation of these, which I will write down because it is important, is 1 to 2 times 10 to the 9. So it's quite a bit. So 1, 2 times 10 to the 9 per kilogram per day. So, which quite a bit. Next, we're going to talk about monocytopoiesis. So it's the same progenitor cell as for segmented neutrophils. The key regulatory factor for this guy is M-CSF. So similar to granulopoiesis. The maturation characteristics are gradual nuclear folding. So if we once again have our stem cell here, and you look as we go down the line here, you might have something that looks like this. And then eventually you have this kind of U-shaped, weird, folded in nucleus like that. It looks like a horseshoe. And then they also acquire cytoplasmic granules in their cytoplasm. And the stages in development would be you have your monoblast, so maybe that might be here. And then you have your promonocyte. And then you have your mature monocyte. And remember that monocytes are called monocytes when they're in the blood or the plasma. And then once they migrate to the tissue and they become activated in the tissue, they're called macrophages. They differentiate into macrophages. So there are specific changes that happen, and that's kind of different than what I'm talking about this video, so I don't want to confuse anyone. So maybe I'll talk about that in a future video. Next, we have lymphopoiesis, which is a little bit more complicated. So lymphopoiesis is the creation of T cells, B cells, natural killer T cells, and natural killer cells. So I will actually write that down. So we have natural killer cells, T cells, B cells, and natural killer T cells here. The regulatory factors, we have interleukin 2, interleukin 3, interleukin 4, and interleukin 7. Now, immunology is a ton of memorization, and there's really no way around it, so it's fun to just create kind of mnemonics. So I just think of the lymphopoiesis as you have interleukin 2, then you have interleukin 3 and 4, and if you add these two together, you get interleukin 7. Just little things like that might help you memorize a little bit better, so it might help. Maybe it doesn't help at all, but you guys can figure out something for yourself to help you memorize these very similarly named interleukins. And then also you have the CD antigens, which I'll talk about in a second. So the stages of maturation of this are actually defined by the expression of CD antigens, because you can't really tell the difference between their morphologies. So it's not like this, and between the different cells you can't really tell the difference between them. One important feature is that B cells and natural killer cells they mature from their progenitors in the bone marrow, right? So the whole thing's done in the bone marrow. When you look at a T cells, or natural killer T cell, they develop in the bone marrow, and then they mature in the thymus. And I'll talk about what the thymus is. But basically, when they're in the thymus, they undergo a lot of complex interactions with different cell types and cytokines, and then eventually they become mature T cells. So they could become CD8 T cells, CD4 T cells, or natural killer T cells. So that's it for the production of this. Obviously, there's homeostasis associated with hematopoiesis, which is just kind of a continuous process that ensures the steady state levels of mature blood cells, which would require, I believe it's 3.7 times 10 to the 11 leukocytes per day. Don't quote me on that. But basically they just control the levels of cells and types of cytokines produced by bone marrow stromal cells. They produce cytokines that have hematopoietic activity. They regulate the expression of receptors, and they remove some cells by apoptosis, which is programmed cell death. And obviously it's important for you to know the difference between apoptosis and necrosis, right? So necrosis would happen when your cells die from something like if you smack your head, or say your hand or wrist, on the corner of a counter, right? You're going to have a big bruise there, and a lot of the cells are died. That would not be programmed cell death. That is necrosis. Necrosis is more of a traumatic cell death. It's not controlled. It's like really, the cell becomes really swelled and leaky, and everything just kind of floats out of it. Whereas apoptosis has very controlled, what's called blebbing, and then that releases apoptotic bodies, and then the nutrients and the biochemicals that are inside those apoptotic bodies can actually be reused for other cells. But yes, that is hematopoietic. The different hematopoietic processes for different types of immune cells.